And we want our sites at Duke and at UK, University of Kansas in Wichita, and a third site in here in Missouri, UK, Missouri, that Missouri, right? That we keep your mics muted, and I hope that you are listening in, and we'll get to you when we have our panel discussion. So our next speaker is Dr. Misha Angrist, who is an assistant professor at Duke University in the Institute for Genome Sciences and Policy. Misha is the author of a book called Here is a Human Being at the Dawn of Personal Genomics, and which has been reviewed quite positively, and was one of the first in-depth looked at personal genomics out there. And Misha has some really keen insights into the personal aspects of genomic testing and some of the important ethical issues. So without further ado, Misha.
Thanks, Rich. It's great to be here. I'm always keen to hear Dr. Kingsmore talk. I could listen to him all day. So, hello, Duke and other remote sites. Morning, ladies and gentlemen, and all the ships at sea. So, I'm going to switch gears, although I'm involved in whole exome sequencing of undiagnosed neonates at perhaps a smaller scale with some colleagues at Duke, led by Nico Katsanis. But I'm going to talk in a way that maybe is unencumbered by data and tell some stories and maybe offer some provocations. And since our raison d'etre here is privacy, I will talk about that. So these are my disclosures. I work for Duke. I'm an unpaid advisor to Invitae, which is a diagnostics company, part of the Personal Genome Project. So you can read about that at personalgenomes.org. You can look at my genome if you're having trouble sleeping. And I serve on the Duke IRB, but I don't speak for it or for Duke. And as part of both my policy research and this ethical, legal, social work I do with respect to exome sequencing that's supported by NIH. So I thought we'd go back, if not to the beginning, to close to the beginning. HIPPO privacy. So the Hippocratic Oath is not only do no harm, but the eighth principle actually says, whatever I may see or hear in treatment or even without treatment in the life of human beings, things that should not ever be blurted out outside, I will remain silent, holding such things to be unutterable. So can everybody see these slides okay? Okay, I'm happy to mess with the lights. So if we move ahead a millennium or so, we come to these two guys, Warren and Brandeis, who finished second and first in their Harvard Law School class in the late 1800s, and they wrote this piece called The Right to Privacy, which has become canonical not only in first-year law school classes, but certainly with sort of respect to the way we view this country. And they talk explicitly about the right to be let alone. Recent inventions and business methods call attention to securing to the individual what Judge Cooley calls the right to be let alone. Instantaneous photographs and newspaper enterprise have invaded the sacred precincts of private and domestic life, and numerous mechanical devices threaten to make good the prediction that what is whispered in the closet shall be proclaimed from the housetops. So perhaps they were anticipating TMZ. So let's jump ahead another century plus. This is a website I just discovered and I'm fascinated by, called Terms of Service, didn't read. And it essentially critiques the privacy policies and the other behaviors of various social media companies as made explicit in their terms of service. So a lot of it's fairly new, so a lot of them don't have official grades. You can see Netflix gets a fair number of thumbs down. Tumblr, no promise to inform you about data requests from governments. This is a recurring theme. Google gets a C. This service tracks you on other websites. Google can share your personal information with other parties. No promise to tell you what the government's up to. Facebook, no grade yet. Very similar. So all of those things happen ostensibly with our consent. And of course, the other elephant in the room that we would be remiss in ignoring is this other stuff that seems to have captured people's attention. This is a New York Times editorial from about 10 days ago. That Chancellor Merkel's cell phone conversations could fall under that umbrella is an outgrowth of the post-9/11 decision by President Bush and Vice President Cheney that everyone is the enemy and that anyone's rights may be degraded in the name of national security. So one of the guys who's done a lot of reporting on the NSA activities and the Snowden leaks is a guy named Barton Gellman, who was actually in Durham a couple of weeks ago and said something that caught my attention. We have become radically transparent to them, and they've become more opaque to us. It's a place where you're almost living inside a one-way mirror. Okay, so you're wondering, what does this have to do with genomics? I ask for your forbearance, but first I want to say that the NSA thing has not been all bad, because we've gotten some absolutely awesome t-shirts and bumper stickers out of it. NSA bugs me, that's pretty good. I like this one. Lost an email number or text. Don't spy on me, bro. Why back up? My data is stored by the NSA. My students like that, and this is my favorite, the only part of government that actually listens. So what can Brandeis and Warren, coupled with NSA, teach us about how we feel about privacy and how we might feel about medical and genomic privacy? And clearly, a lot of this has to do with technology and the ability to get access to information about people that in many instances they would rather we didn't know about. So this is a slide which some version of, probably many or most of you have seen. NIH actually has a website devoted to the falling costs of DNA sequencing and updates it regularly. And what you see here on the blue line is Moore's law, that is the number of transistors per chip, basically the amount of computing power per dollar that we can get as a function of time. And the orange is our ability to sequence DNA, to decode DNA, the types of stuff that Dr. Kingsmore spoke about so eloquently. And you can see that we're approaching 10 to the 11th bases per person per day, something like that. And in the yellow, you see actually our ability to write DNA, which has given rise to the whole field of synthetic biology. which I won't talk about, is also growing exponentially. So this technology is cheap and getting cheaper. And so we have to think about some of the issues that Dr. Kingsmore discussed and the broader notion of how we feel about this information and what we want to do with it. But I'm interested not only in sort of the organic issues as raised by somebody, say, getting a diagnosis and having access to millions or billions of bases of incidental information, but how we sort of preemptively choose to interpret this stuff. So I won't belabor the HIPAA privacy rule, just to remind you that it is about protected health information, things like your name, social security number, address, email address, and how the regulatory system as it stands places a premium on this sort of information and how careful we are expected to be with it. The HIPAA security rule is essentially an outgrowth of that idea, but it is specifically meant to govern how we treat online information, electronic PHI. And I bring that up because this is another paper that caught my attention that appeared, I think, in the American Journal of Public Health earlier this year. And I love this paper. And it's about some public health workers' desire to communicate information about influenza vaccination to their audience, to a public health audience. So the idea is simply some kids require a second dose of flu vaccine 30 days after the first one. To send influenza vaccine reminders to parents, we wanted a simple direct text message that all parents would understand easily and that would not trigger the security rule. So they thought, how can we do that? So this was their first attempt, and the message was, it is time for insert child's name, second dose of seasonal flu vaccine. Visit a pharmacy or clinic today for the booster to keep your child protected. Well, that's not going to fly, they said, because if this message were sent to the wrong person, the unintended recipient would know who the child was, and the reference to the need for the second dose would be considered PHI. Okay, let's try again. It is time for your child's second dose of seasonal flu vaccine. Visit a pharmacy or clinic today for the booster to keep your child protected. It's essentially the same as the first, because presumably someone who recognized the number could discern the child's identity. Okay? How about this? If it's been 30 days since a first flu shot, then it's time for some children to get a second dose of flu vaccine. Call a doctor or pharmacy to schedule an appointment. This is preferable from a risk management standpoint, but unacceptable from a communications perspective because of the vagueness of the message. Okay, finally, double whammy. Keep your child protected against the flu. Some kids need a second dose 30 days after they receive their first flu shot. Message number 2, do you remember asking for a text message reminder for flu vaccine? It's time. Call a doctor or pharmacy. So this avoids the use of PHI and is, quote, closer to our goal for clear communication. I don't know why this strikes me as so comical and maybe tragicomic, but this is where we are with message about flu vaccines with respect to medical privacy. Okay, so now to switch to genomic privacy, Dr. Kingsmore mentioned the incidentalome. All of this information, obviously, we count social security number as PHI. It is a 9-digit digital code. Our genomes are a 3 billion or 6 billion digit digital code, so that's hard to protect. So this is a paper by a guy named Yaniv Erlich from the Whitehead Institute that came out early this year, which uses a combination of publicly available genomic information that has ostensibly been de-identified, coupled with publicly available genealogy information. And in fact, this exercise was accomplished first in 2005 by a young man who was the child of his mom and an anonymous sperm donor, and used a combination of a Y chromosome genomic test coupled with some clever genealogical searching, and I think maybe he had a surname, and re-identified his ostensibly anonymous biological father. So what are the implications of this? Ehrlich's study upped the stakes because it showed that it was possible to identify people from their genomic data, genetic data, by linking not to other sources of research data, but to information freely available on the internet. So people who have agreed to participate in genetic databases like the HapMap, like the 1,000 Genomes Project, and have done so under the presumption of being anonymous, are probably at some level not anonymous. Okay, another story. We may ask patients and participants what they want with respect less about privacy, but about our relationship with them. But it's not clear whether we're really interested in the answer, and I will make the argument that this actually has some bearing on privacy, and maybe we're using privacy as a bit of a fig leaf or an excuse. So the Million Veteran Program was, I think, launched, or the genesis of the idea came a couple of years ago. You know, veterans are an extremely well-tracked cohort. They have their own healthcare system. It has its own research infrastructure. And so it seems like a wonderful opportunity to learn about genomics and hereditary factors and perhaps health outcomes that are peculiar to veterans, post-traumatic stress disorder, exposure to various chemical agents, that sort of thing. And so this led to the MVP. And the MVP is designed to be essentially a large genomic biobank. And to its credit, the MVP employed the Genetics and Public Policy Center to conduct a survey to ask potential participants, would you be interested in this? What are your expectations? And lo and behold, veterans said, 2/3 of them said, they would be less likely to join if they did not get research results back. And 90% said they would want to know all of their research results. Okay, seems fairly clear. On their website, under the FAQ, the MVP says it will not be possible to give participants results of the blood test due to regulations under the Clinical Laboratory Improvement Amendments. We are legally unable to return research results to participants. Now, we can argue about what the meaning of the CLIA statute is and the legitimacy of that excuse. I'm just making the point that here was a research enterprise relying on the goodwill of veterans. And if you go on the website, you can watch all sorts of video testimonials from veterans about how the MVP is giving back to them. The MVP asked veterans what they wanted. The veterans said, this is what we want. And the MVP said, well, we're sorry. Next story. We occasionally do the right thing, but even when we do, the asymmetry between doctors and researchers and physicians and patients remains. So I'm not, Rich alluded to it, I'm not going to get into what I see as the increasingly arbitrary distinction between research and clinical care. But of course, no talk about ethical, legal, and social issues in genetics is complete without talking about Henrietta Lacks, who, as most of you know, was a poor African-American tobacco farmer. She was diagnosed with an aggressive form of cervical cancer in 1951. She eventually succumbed to that disease. Hers were the first human cells to be widely propagated in culture. And her cells have been a staple in the laboratory for 6 decades and have been part of all sorts of amazing discoveries, although the Lacks family was never told what was being done with their cells, and they certainly did not benefit financially. Rebecca Skloot has written this wonderful best-selling book about it, and this is a small piece of the story that I'm especially captivated by because I think it sort of prefaces where one of our deficits lies with respect to research participants and perhaps patients, and particularly what's gone on in the last few months with the Lacks family and NIH, which I'll get to. So Christoph Lengauer, who was a postdoc at Johns Hopkins, read a piece that Rebecca Skloot wrote about Henrietta Lacks in the late 1990s and reached out to her and said, I would like to invite the Lacks family to come to my laboratory. I want to tell them a little what HeLa means to me as a young cancer researcher and how grateful I am for their donation years ago. I do not represent Hopkins, but I'm part of it in a way I might even want to apologize. So that was, I think, in the year 2000. And so we jump ahead now to earlier this year, and a group from the European Molecular Biology Laboratory publishes the genome sequence of the HeLa cell line. And not surprisingly, as with most cell lines that have been passed for countless generations, the HeLa cell line has all sorts of genomic rearrangements in it. But for whatever reason, it did not occur to these researchers to approach the Lacks family or let them know what was going on. In this paper about Yaniv Erlich, he comments, or they cite a tweet that he posted after Embl said that the genome sequence published for the HeLa cell line would not reveal anything about Henrietta Lacks or her descendants. And he simply said, nice lie, Embl. And Rebecca Skloot wrote about this for the New York Times and quotes Jerry Lacks Y, Henrietta Lacks' granddaughter, who said, that is private family information. It shouldn't have been published without our consent. So this precipitated several things. One is the Embl researchers took the HeLa genome down from public databases. Meanwhile, in this country, Jay Shenduri at the University of Washington was getting ready to publish his own analysis of the HeLa genome in Nature. And because he was NIH funded, Francis Collins and Kathy Hudson from NIH said, well, this seems like an opportunity to meet with the Lackses, which they did. And they reached an accord that led to the HeLa data being deposited in a restricted access database. And this agreement specified that the HeLa data would be used for biomedical research only. Researchers would not initiate contact with the family. They would be obliged to disclose any sorts of commercial plans. And they would acknowledge the Lax family in any HeLa-related publications or presentations. And so this appeared in the same issue of Nature in August describing this deal. And you can see here that it says the genome described in this paper was derived from a HeLa cell line, Henrietta Lacks, and the HeLa cell line that was established from her tumor cells in 1951 have made significant contributions to scientific progress and advances in human health. We are grateful to Henrietta Lacks, now deceased, and to her surviving family members for their contributions to biomedical research. So this is essentially what Christoph Lengauer had done of his own volition in 2000, many decades after this first happened. So this is from that discussion by Hudson and Collins, and this is a picture of the Lacks family next to a historical marker dedicated to her in Virginia. But one of the things they say in that paper is, look, we are not making a template here. This is an extraordinary case. We are not setting a precedent for research with previously stored de-identified specimens. This is unique. And around the same time, say a month later, the Department of Health and Human Services issued a new or an updated draft NIH genomic data sharing policy and asked for public comments. And so what we see here is the NIH expects the informed consent process and documents to state that a participant's genomic and phenotypic data may be shared broadly for future research purposes, and also explain whether the data will be shared through open or controlled access. So perhaps the Lacks process has influenced this. And the NIH expects that informed consent for future research use and broad data sharing will have been obtained, even if the cell lines or clinical specimens are de-identified. So perhaps an acknowledgement that true de-identification is hard to do, if not impossible. But again, the expectation is that informed consent will do this work. And for various reasons I won't get into, I'm somewhat skeptical of that. I think informed consent has a lot of problems. So let me finish by saying I think what Barton Gellman said about Google and Facebook and the NSA, we might say about genomic research, that on some level, those of us who participate in this research and share our genomic information with researchers have become radically transparent to the people studying us. Clearly, research participants value privacy. That's absolutely true. Even though most of us, it seems, have agreed to exchange privacy for other stuff that we get from places like Google and Facebook. I think it's clear that certainly with respect to genomic information, it's very hard to make any sorts of guarantees about privacy and confidentiality. And I think we pay a price when we sort of structure our relationship to research participants with privacy at the top of the pyramid, perhaps at the expense of other things, one of which I would argue is communication. There are places, there are studies, people like Mike Baumschad and Holly Tabor at the University of Washington, people like Sharon Terry at Genetic Alliance, who are initiating an ongoing, abiding, open, two-way exchange of information with the people they study. They believe that value that aspect of the research, and the relationship with their participants is more important than trying to honor the letter of HIPAA privacy. So I will leave you with Ralph Ellison. Thanks.
Thank you. Thank you, Misha. Some very intriguing observations and comments there that we're going to probe a little bit later.